Hello, everyone. Welcome to today's Society of Neuroscientists of Africa webinar series. My name is Dr. Mahmoud Bukarmena. I'm a research fellow in the University of Sussex Neuroscience, and I'm one of the co-organizers of this webinar series. And I'm very excited to say that today we have Professor Louise Serpell from Sussex Neuroscience, who's going to give the webinar. Professor Louise Serpell is a professor of biochemistry at the University of Sussex, UK, and also the Director of Sussex Neuroscience. Louise is a leading expert in the role of protein misfolding in neurodegenerative diseases. So Louise started her work on amyloid fibril structure during her PhD at the University of Oxford in the UK, before moving to the University of Toronto in Canada in 1995 to focus on Alzheimer's disease at the Centre for Neurodegenerative Diseases. She then returned back to the UK to the Laboratory of Molecular Biology, that is the MRC Centre in Cambridge in 1997. And then she took up a fellowship at the University of Cambridge in 2000. In 2003, she joined the University of Sussex, where she became a full professor in 2010. So Louise's work focuses on protein self-assembly and misfolding at the heart of neurodegenerative diseases and amyloidosis. She has worked on protein misfolding for over 25 years and published over 140 peer-reviewed articles and holds one patent. She's credited with developing a genetic structural model for amyloid fibrils, and her work has been funded by the Wellcome Trust, BBRC, MRC, Alzheimer's UK, Alzheimer's Society, as well as TAUREX. She is frequently invited to speak at international conferences and elected to organize numerous key meetings in protein misfolding and neurodegenerative diseases. She sits on the board of many grant panels, as well as serving as the reviewer for many journals. She is also the co-director of the Alzheimer's Society Doctoral Training Center in Sussex, which focuses on the role of ApoE as a risk factor for the late onset Alzheimer's disease. Louise is a co-founder of the Dementia Research Group in Sussex, and her work currently spans from structural biology to cellular neuroscience to uncover the mechanisms that lead to neurodegeneration in aging and diseases. And I have to say that in addition to all this, she is an excellent mentor and science communicator. She regularly organizes public engagement activities to inspire the next generation of neuroscientists and promote public understanding of dementia. And in fact, for those of you who probably know me from Africa, organizing activities or leading some research programs, it is due to her mentorship and support for the mentees in her group that I am able to do all these activities that I've been doing. So it's really, really exciting to have her today, and for her role for her research as well as mentorship roles, she has been recognized on multiple occasions. Louise, is very, very, it's a huge pleasure to have you today, and the stage is all yours.
Thank you so much, Mahmoud, and thank you to Mahmoud and to Mossab and all of Sonar for organising this today. So this is the first time I've done something like this, so bear with me. So I'm first of all going to open the slide presentation, and hopefully, Mahmoud, you'll just let me know if there's any problems as I'm talking. So what I want to talk to you today about is our perspective on looking at neurodegeneration in Alzheimer's disease. So as a protein structural biologist for a long time in my career, we look at things in a very protein-centered way. And what you can see here in this very first slide is some paired helical filaments that are formed by tau in Alzheimer's disease. So what I'm going to do is to do an introduction. I'm going to talk to you a bit about protein misfolding for those of you who don't know too much about it. So in this next slide, what I'm showing here is the native structure of a protein shown here. This happens to be a protein called lysozyme, but it's not it could be any protein, as you'll come to see as I speak. We have a protein chain that starts to self-assemble and form an oligomeric species, so a bundle or a group of proteins. These are identical proteins. They then go on to form a protofibril, and at this stage they become a little bit structured, and then we get the amyloid fibril, which I'll talk a lot about, and finally this is a deposited amyloid plaque, so lots of amyloid fibrils in an amyloid plaque. And while I was speaking, what you maybe saw was this little movie. This was done by Carol Hall some years ago, And what this little movie showed was if you take some individual peptides and you put them into a box, so this is a computer simulation, and you just allow them to sort of move around, what you find at the end, and you can see that here, is that they start to bundle together, organize themselves, or self-assemble to form these very ordered structures. And the main thing that we need to remember about amyloid is that it's a very ordered structure. So what is amyloid in terms of disease? Well, I've shown a picture here. This is an electron micrograph showing amyloid fibrils formed from a particular protein. In a way, it doesn't matter what that protein is, because many different proteins can form amyloid fibrils. And this can create some confusion sometimes in the field, because in Alzheimer's disease, those of you who work on Alzheimer's disease will know that there's a protein called amyloid-beta. Amyloid-beta is shown in this table. Amyloid-beta, or A-beta, is a short peptide, 40 or 42 amino acids long, and it forms amyloid fibrils in Alzheimer's disease. Also, though, tau also forms amyloid fibrils in Alzheimer's disease, just to confuse you. And amyloid is defined as a very specific structure. So it's not that amyloid itself is amyloid beta, but amyloid is defined as a much broader term. So, for example, in Huntington's disease, in diabetes type 2, in Parkinson's disease, and in amyloid light chain disease, we have the deposition of a particular protein that forms amyloid fibrils, and it accumulates in the tissues. So these are not all neurodegenerative diseases. They're not all forming amyloid in the brain. So in diabetes type 2, this protein, islet amyloid polypeptide, forms amyloid fibrils, and they're deposited in the pancreas, whereas the amyloid light chain can deposit in lots of different places. This is formed by the part of an antibody, immunoglobulin light chain. And in the next picture, I can show you the deposition of amyloid in the heart of a patient who has this amyloid light chain disease. So on the left-hand side is a normal heart, and on the right-hand side is the heart of a patient with heart failure. And the reason that they have heart failure is because of the deposition of this amyloid fibrils formed from immunoglobulin light chain. So I want to talk to you a little bit about the structure of amyloid and what makes something amyloid. So it turns out that amyloid fibrils, despite having been formed by many different proteins, is made up of what we know as a cross-beta structure. And we know that from a number of different observations over the last 50 or 60 years. And I've just shown you a few examples from my own lab here. So this is an electron micrograph, actually a cryo-electron micrograph, showing a short peptide of amyloid beta that forms amyloid fibrils. And we can see in this cryo-electron image these striations across the fiber axis that are 4.7 angstroms apart. So this will become important when we start to look at this little movie here. We also, in the past, have used X-ray fiber diffraction to look at the structure of amyloid fibrils from lots of different amyloid fibrils, and we always get this very specific X-ray fingerprint. What the X-ray structure diffraction pattern shows you is, again, 4.76 angstroms on the meridian. So this is a reflection coming from the repeating units along the fiber axis, and then usually a more diffuse reflection at about 10 angstroms. So hopefully this will become a bit clearer when I show you this model. So this structural model was built by Kyle Morris, who was a student and postdoc with me a few years ago. What you'll see in a moment is where those reflections come from. So here we have the structure of the amyloid fibrils, so the model structure. And what hopefully you can see in a moment is the beta strands that are depicted by the arrows, the blue arrows, running perpendicular to the fiber axis, and these hydrogen bonds that hold the whole structure of the amyloid fibril together. So this gives it its incredible stability. And now if we look down the fiber axis, what we see is the side chains of the protein interdigitating to form two beta sheets that are organized to form the amyloid protofibril. So what you will have seen here is this cross-beta structure, this generic cross-beta structure that can be made up of numbers of different sorts of proteins. So this is the cross-beta structure. So I'm going to move now on to what Alzheimer's disease actually is. So this is a very general slide, pointing out that Alzheimer's disease is just one form of dementia. So Alzheimer's disease is around 55% of all dementia cases, but other dementias, of course, do exist, including vascular dementia and frontal temporal dementia, and then many other types of dementia that are found in the population. So what causes Alzheimer's disease specifically? Well, there are some genetic reasons for people getting Alzheimer's disease, and we can have mutations in the amyloid precursor protein, and this protein is a large transmembrane protein which is processed to make our amyloid-beta peptide. We can also have mutations in two proteins called the presenilins, presenilin 1 and presenilin 2, and these proteins are involved in the machinery that actually generates A-beta from the amyloid precursor protein. And finally, people who have Down syndrome, where they have trisomy of chromosome 21, will have an overproduction of amyloid beta. And this is because the APP, or amyloid precursor protein, gene is found on chromosome 21. So what you might have noticed from this genetic explanation of Alzheimer's disease, that A-beta comes up a lot. It seems to be a really important protein in terms of the cause of Alzheimer's disease. So there are also some genetic risk factors. There are two genes that can encode Apolipoprotein E2, E3, or E4. So people can have E2, 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 E3, 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 and so on. And it's been shown that people who have E4, E4, so this is a polymorphism, have an increased risk of developing Alzheimer's disease at an earlier time, but generally over the age of about 70 or 80. So these are genetic factors, but they don't mean you will definitely get Alzheimer's disease. They mean you have a higher risk. The most important cause of Alzheimer's disease seems to be aging. So we know that aging leads to the decline in protein control and protein folding mechanisms. It increases oxidative stress. And of course, as we go through our lives, we might accumulate damage from environmental impacts, whether those might be from disease or from pollutants and so on. But what about tau? So I mentioned right at the beginning that tau forms paired helical filaments. And in Alzheimer's disease, we have both amyloid plaques and also neurofibrillary tangles made of tau. And tau is interesting because not only is it involved in Alzheimer's disease, but it's also involved in a number of other diseases known as the tauopathies. So these include frontal temporal dementia, progressive supranuclear palsy, chronic traumatic encephalopathy, and cortical basal degeneration. So these are all different diseases that involve tau, but not A-beta. So tau is obviously a very important neurodegenerative disease causing protein. So we can't put that aside and ignore it in terms of Alzheimer's disease. So now I want to connect the dots between what I talked about in terms of structure and what I've talked about in terms of Alzheimer's disease. So this is an image that probably you've seen many times. On the left-hand side is a healthy brain, and on the right-hand side is an advanced Alzheimer's disease brain. So the patient that has a brain that looks like this will have a huge amount of neurodegeneration and shrinkage of their brain tissue. And if we were to look inside this brain, what we would see is amyloid plaques. So this shows you a light micrograph showing amyloid plaques, a histopathology section. And so you can see this densely labeled structure, which is the protein structure within, and the dead or dying neurites on the outside surrounding this amyloid plaque. Of course, this is quite low resolution. So if we want to look in a bit more detail, we can use an electron microscope to look at the amyloid fibrils that are formed. So this is some electron microscopy that was done using amyloid fibrils that are formed in vitro. And what you'll notice is that they're very, very long fibers. They can vary slightly in their morphology. So you might see there are some quite straight filaments, some quite helical or twisted filaments, some that are quite wavy, and so on. So within our sample, we might get different sorts of filaments. But what they all share, most importantly, is that they are cross beta. So if we look in a bit more detail into the structure of these, so we zoom in even further, what we see is the beta strands running perpendicular to the fibre axis, as I showed you in that little movie earlier on, hydrogen bonded all the way along the length of this fibre, and forming this very highly repetitive structure. So there are several protofilaments that are each formed of these cross beta structures. And what's interesting about this structure is because it's so repetitive, we can access it using many different types of protein. So if I show you the structure of amyloid beta here, it's just a little hairpin, and it can stack up on itself, self-assemble to form this paired, this cross beta structure. But if I was to take a larger protein, for example, I showed you at the beginning something like lysozyme that forms amyloid fibrils in disease, then I would need to fold it more frequently in order for it to access this structure. But I could do that because this is a very repetitive structure. So I'm going to focus completely now on Alzheimer's disease, and I just want to show you in this slide the comparison between the two hallmarks of Alzheimer's disease. So on the left-hand side, we have a light micrograph of showing these amyloid plaques, and this is an electron micrograph showing the amyloid plaques in a bit more detail. So this is an electron micrograph showing you the amyloid fibril deposits in the brain tissue of a patient. And these are some amyloid fibrils that are formed in vitro using this protein amyloid-beta. So these are the amyloid plaques formed from amyloid-beta, and they are extracellular, so they're outside of the neurons. If we look on the right-hand side, we see the neurofibrillary tangles that are formed in these little flame-like structures within the cells. So these are intracellular. If again we look at this using electron microscopy in the tissue of a patient who had Alzheimer's disease, you'll see that these are paired helical filaments, and the little dots you see are gold labels that we used to label the tau deposition in his brain. So we knew what we were looking for. So the dots are not part of the paired helical filaments, they're just gold particles to help us find them. And if we look in a bit more detail, we can actually see why these are called paired helical filaments, because they have this very precise paired helical morphology. And we can make these in vitro. And I'm going to tell you a bit more about that towards the end of this lecture. So these proteins are formed from tau, they're intracellular, and they're known as paired helical filaments, which are formed within neurofibrillary tangles. So I'm going to start off by talking a little bit about some work that we've been doing on amyloid beta. So the key question is really, what are these proteins actually doing? How might they be involved in the cause of Alzheimer's disease? So this is amyloid beta, and again I'm showing you this assembly profile from monomer to aggregate to oligomer to protofibril to amyloid fibril and to amyloid plaque. And the question is, how is this process, or these amyloid plaques perhaps, causing neurodegeneration? How might they be affecting neurons? So this is a very simple diagram of a neuron. And what it just reminds me to point out is the parts of the neuron that might be really important for the health and the function of the neuron. So of course all cells have mitochondria that produce the energy, and they all have lysosomes where proteins are degraded, regenerated, amino acids need to be regenerated to make more protein. And this is the nucleus here. In a neuron, of course, it's very important that transport of cargo is facilitated along the very, very long axons. And particularly, we need to consider how various cargoes are delivered to the synapses so that the function of the neuron can be maintained. So all of these parts of the neuron are important. And it turns out that amyloid beta has an effect on many parts of this function of a neuron. So many people have reported on effects on mitochondrial function, on lysosomal function, on transport of cargo, and on synapses. And what might bring this all together is that each one of these is surrounded by a membrane. So we have the plasma membrane, of course, and then membranes that surround the synaptic vesicles and the lysosomes and the mitochondria and so on. So the first thing that we did quite a long time ago now is to look at the effects of A-beta on biomimetic membranes. So the idea of this was to recreate some membranes that are similar to those found in cells in the test tube, and then to examine what effect A-beta has on them. So what's shown here is a little lipids bilayer, large unilamellar vesicle, and within this vesicle are some fluorescent molecules. This is actually called calcine, and it's a self-quenching molecule, which means that when it's within these vesicles, it doesn't have any fluorescence. So what we can do is add amyloid-beta and see what effect it has on these vesicles, and we can measure that using fluorescence. And what we find is that the fluorescent molecules are able to leak out of the vesicles because the amyloid-beta affects the permeation of the vesicles. And when these leak out, they become fluorescent, so that we can actually measure that fluorescence and we can measure the permeation of these vesicles. And then we can have a look and see which part of the assembly process is most important for causing this membrane permeation. So we incubate the amyloid-beta for different lengths of time. So here we've shown 1 hour, 5 hours, 24 hours, and 72 hours. And what you see is that we get these little oligomeric species early on, and then we start to see some fibrils and some protofibrils. Finally, we start to see these fibrils, and at 72 hours, we see lots and lots of fibrils. And what you'll notice from this graph here is that as we increase the size of our molecules, as we increase the elongation of the fibrils, we get less and less permeation of the membrane. So we get most fluorescence leaking out when we have very low, small molecules, and much less leaking out when we have these large fibrils. So what this shows us, as other people have shown, is that the oligomers of amyloid-beta seem to be very important in terms of causing damage to membranes. And we can look at that damage a little bit more using a method called atomic force microscopy. And what this allows you to do is to visualize the height of molecules. So shown here is a bilayer, and it's relatively flat. And you can see that because the colors are dependent on the height of the molecule above the surface. This is actually an electron micrograph, which shows you the structure of the oligomers that have been added, the amyloid-beta oligomers. And on the right-hand side, you can see what happens to this flat membrane when we add amyloid-beta to it. So the important thing to note about this is that when the colour is dark, it means it's lower than the surface, and when it's bright, it's higher than the surface. So atomic force microscopy gives us height information, surface information, and what we can see here is what appears to be holes in the membrane. So we can see little fibrils forming, and you can see that in a bit more detail down here. So we can follow these forming on the surface, and then we can also see these very dark structures, which seem to be holes in the membrane. So we can actually see that damage being, occurring on these membranes. And this shows you in a bit more detail, so you can see more information here. So of course, this is all very in vitro. These are biomimetic membranes. So the next thing we want to do is to look at what happens when we add amyloid-beta to cells. So we're going to look at cytotoxicity here. And this image shows you some hippocampal neurons that have been grown in culture. And you can quite clearly see these neurons. You can see their cell bodies and their neurites and dendrites and so on, forming a nice network of neurons in our plate. And what we can do now is we can use a toxicity assay to look at how many of these cells die when we add A-beta and how many of them survive. So we can colour all of those, all of the nuclei in blue using DAPI. And then we can identify the cells that are dying in green here to measure the amount of cytotoxicity that is occurring. So that's how we do our assay to see the effects of amyloid-beta. So before we do that, we have to think about one really important thing. We haven't got a control. So we decided that we would prepare a control for amyloid-beta. So I'm going to tell you a bit more about that now. So we decided to rationally design an amyloid-beta variant that cannot assemble. So let me just take you through this slide a little bit slowly. So this is an algorithm that was written by Joost Shimkowitz and Fred Rousseau, and it's called Waltz. and it's actually available online. I can provide the link if anybody would like to use it. So essentially what you do is you put in your sequence, and it will run through that sequence with 6 residue windows, and it will predict how likely it is to form an amyloid fibril. And so what is shown here in this table is when it's red, it's unlikely to form amyloid, and when it's green, it is likely to. So you can basically get a score for a structure, and you can get an output for amyloid beta that's shown in this graph here. So essentially what this shows is that there are regions of amyloid beta, this is the sequence of amyloid beta 42, where it forms amyloid, it seems to be responsible for the amyloidogenicity of the peptide. So you can see these 2 regions here that seem to be important in the amyloid forming propensity of A beta. So what we did was we introduced some mutations into the sequence to see if we could remove that amyloid propensity from the sequence. So we introduced a serine for a phenylalanine at position 19 and an aspartic acid for a glycine, sorry, at position 37. And so what you can see here is are sort of ideas about how these peptide, these positions in the peptide might be important. So this is a structure suggested for amyloid beta, and you can see that phenylalanine 19 and glycine 37 seem to play very important roles in the sort of network that's produced within fibrils and within oligomers. So that's the idea. So the first thing I want to show you is just to remind you that amyloid beta forms oligomers, and they grow and they form amyloid. They form these protofibrils, fibrils, finally very dense fibrils, and lots of fibrils. If we take our variant A beta that we then made, we will find that it doesn't form fibrils. I also want to explain this, which is we use thioflavin T fluorescence, which allows us to follow the assembly of the fibrils. As the fibrils assembly, the thioflavin T molecule starts to fluoresce. And we also use this technique called circular dichroism that allows us to look at the secondary structure in solution. So when we first make the A beta, it's random coil, and that's shown by this very specific signal that we see at 195 nanometers by CD. And as the A beta starts to become more and more beta sheet rich, it starts to shift so that we get a signal at around 218 nanometers. So you can see that here, this trough. So what I'm showing essentially is the move from a random coil structure to a beta sheet structure. As I've said, amyloid fibrils are beta sheet rich. So when we do exactly the same experiments with variant A beta 1 to 42, it doesn't assemble at all. The thioflavin T is completely flat, so there's no fluorescence. The peptide remains completely random coil by CD, and by electron microscopy, we don't see any fibrils at all. So we've successfully made a potential control for amyloid beta method. So this is a summary of that data. We show that amyloid beta doesn't assemble, and we've also got some gels here that show large molecular weight structures for A beta and none of those for variant A beta. We show that it's not toxic, so this amyloid that has this non-amyloidogenic peptide is not toxic to cells. So you can see here, this is amyloid beta and this is variant amyloid beta. So you can see the variant amyloid beta is not toxic, and also it's not internalized into cells. So you can see here that the A beta 42 is internalized. This is tagged with a red fluorescent tag, whereas the variant A beta is not. And finally, I'm not going to say very much about this, but we did some assays looking at memory loss in snails, and we found that the A beta 1 to 40 causes memory loss in snails while the variant does not. So you can ask me some questions about that if you like later on. So then we wanted to look at what happens to the amyloid beta. And so this is a little bit more technical. What we did now was to take the amyloid beta oligomers and to fluorescently label them with a particular dye. This is called Cypher, and it fluoresces in acidic compartments. So first of all, I want to show you a little movie showing just an Alexaflor. So this is a green fluorescent tagged A-beta, and you can see that A-beta being internalized into the cells. So we know for sure that when we add A-beta, it gets into the cells. And when we add this cipher, it gets brighter and brighter as it's internalized into acidic compartments. So as we go through the early endosome to the late endosome to the lysosome, we get more and more acidic. So we can actually follow that. And we find that the oligomeric A-beta internalizes into the lysosomes and co-localizes with a lysotracker marker. So we use this lysotracker marker, which is red, the amyloid beta, which is green. And if we look at the co-localization, we can see that they're both in the same compartments. So we know that the amyloid beta is getting into the lysosomes. If we use the cipher tagged A-beta, we can actually follow the kinetics of that internalization. So we actually can follow that over time, and we see the A-beta 42 cipher going into the cells, increasing in fluorescence, whereas the variant A-beta 42 does not get internalized. So you can see that quite clearly here. And this is depicted in the images below, but this is the analysis of that data. What's really interesting now is if we consider what actually is happening to the cells, what effect is the amyloid beta being inside those lysosomes actually having on the cell function? So in order to do that, we used a protocol that had been shown before by the Radford group, and what they did was to take ovalbumin, which is a protein that's very easily internalized, and this time we've tagged this with the cipher probe. So we can follow its internalization into the lysosomes just like we did with the A-beta. So you saw that in that little movie there. It gets brighter and brighter as it gets internalized into these acidic compartments. And so now this means that we can incubate our cells with A-beta, and then we can look at what happens to the ovalbumin internalization with time. And what we see is that when we incubate only with buffer, the ovalbumin gets internalized really nicely. You see this brightness. But when we have pre-incubated with amyloid beta, we don't really get very much internalization at all. So what we're showing here is that the amyloid beta is able to impair the endolysosomal pathway, so the internalization of other proteins. So that might be really important in terms of its toxic effect. So to summarize, we show that oligomeric A-beta impairs membranes and it's neurotoxic, and it internalizes and causes dysfunction in the endolysosomal pathway. So now I want to switch on to tau and talk a little bit about tau. So all I want to say really regarding this slide is that tau is complicated. It's not just one peptide, it's lots and lots of different proteins. Shown here, all the different types of tau that can be formed. There are 3 repeat taus, there are 4 repeat taus, there are tau with different N-terminal regions and so on. So there are quite a lot of different sorts of tau. Tau has been shown to bind to microtubules. It's also found in the nucleolus, and that was actually Mahmoud's work showing that it's associated with ribosomal DNA in the nucleolus. It can be non-phosphorylated, phosphorylated, or hyperphosphorylated, and it can also be truncated. So what I want to show you in the last few slides really is the work that we've been doing to look at the structure of tau paired helical filaments. So tau causes a little bit of a headache because if you take full length tau, it does not self-assemble and form paired helical filaments in vitro unless you add an additive like heparin. And so we started to work on a region of tau, which is shown here, 297 to 391, and that covers the repeat 3 and repeat 4 region of tau. And so if you notice in the previous slide, that's common to both 4 repeat tau and 3 repeat tau. So 3 repeat tau has R2 missing. So it's a nice model system to work with. This arose from some work that was done a long, long time ago that showed that an antibody that's raised against this final amino acid, the 391, labels paired helical filaments in vivo. and that's shown here. So these are gold particles that label the paired helical filaments using this antibody that identifies this truncation. And more recently, some structural work was done by the Godet and Sherez groups, where they showed that the structure of paired helical filaments from Alzheimer's disease brain have a particular core within them. And that's shown in this picture here. And it turns out that that core is sequence 306 to 378. So that fits nicely into our 297 to 391 region. So we're actually looking at something that might be important in the core of the paired helical filaments. So what we're doing here is looking down the axis of the fibre. So the fibre would be coming out towards you. So I just want to show you that if we use this short peptide 297 to 391 that we sometimes call DJE, as you'll see it's labelled here, it self-assembles without additives to form cross-beta paired helical filaments. So we can use thioflavin S fluorescence, which again allows us to follow fluorescence with time. And we can also use this CD, circular dichroism, to see the loss of random coil as the filaments start to form. And if we spin our sample to separate the supernatant from the pellet, so the pellet is enriched with fibrils, we see that it has a beta sheet rich structure. We can also look at these under the electron microscope. They look like paired helical filaments. We can do some X-ray fiber diffraction and they give a cross beta diffraction pattern. And we can look at these in a bit more detail and analyze their structure. And so we did that, and we compared the paired helical filaments that we'd made in vitro with those that are found in brain tissue. So these are actually in situ in brain tissue. We used an antibody called T22, which actually binds to oligomeric A-beta, oligomeric tau, not A-beta, oligomeric tau. And so you can see these paired helical filaments, and we were able to compare our paired helical filaments formed in vitro with those that are found in vivo in the brain tissue. We compared them, their crossovers and their diameters, and we used also atomic force microscopy in collaboration with Wei Feng Zhu at the University of Kent. And you can see these beautiful paired helical filaments here in the AFM. So this now becomes a really useful tool because it means that we can recreate something like the paired helical filaments that have formed in people, but in a test tube. And that means that we can test potential compounds. And so that's just what I want to show you in the last few slides. We've been working with a company called TauRx Therapeutics that are based in Aberdeen to look at a drug that has been established to treat Alzheimer's disease. So this is a drug that is currently in phase 3 clinical trials for Alzheimer's disease. It looks something like this. So it's derived from this molecule. It was originally derived from methylene blue, and it was a long time ago shown that it can have an effect or a damaging effect on paired helical filaments from AD tissue. And this molecule is interesting because it forms MT+, which is a blue colour, and LMT, which is completely transparent and not colourful at all. And under reducing conditions, it forms LMT. Under oxidizing conditions, it forms MT, and that becomes quite useful when we're looking at the mechanism of action. And so all I want to show you here is that using circular dichroism, we can follow the reduction of random coil over time. And we can follow the increase in beta sheet over time. And so what we're looking for here is the compound being able to prevent that transition to beta sheet structure and the loss of random coil. So what you can see here is that when we start to add some of the LMT, so this is reducing conditions, so we know it's LMT, we start to see less beta sheet and more random coil. So you see this is one without any LMT, this one has increasing amounts of LMT and so on. So when we get to the orange one, we see we now have no beta sheet left, we have lots of random coil left here. We can also see by using SDS gel electrophoresis and then separating the supernatant from the pellet that to start with, when we have just control, we have lots in the pellet. And as we go in increasing the LMT amount in the ratio, all of the tau starts to be found in the supernatant and not in the pellet. So again, that supports our view that this is preventing self-assembly. But of course, the main thing that you want to do is to actually see these filaments. And so we can use electron microscopy to look at these filaments under the microscope. And so this is our control with no LMT at all. And as we increase the amount of LMT, you see that we've got a reduction in these filaments. And so this shows that the LMT is able to prevent the formation of paired helical filaments in vitro. So to summarize what I've talked to you about today, the process of assembly of A-beta from monomer to fiber produces toxic assemblies. We've shown that, but many other groups have too. We introduced a new variance control peptide, which has 2 amino acid changes that render the A-beta non-fibrillogenic and non-toxic. We've shown, and others have, that oligomers can enter cells and accumulate intracellularly in endosomal and lysosomal compartments, and we've shown that there's an impairment of the endolysosomal pathway. And I also showed you a new model system, the truncated tau, that forms paired helical filaments, which may seed endogenous tau, so that might be something we could look at in cells, and that this tau assembly of this short peptide can be inhibited by this compound LMT. So that might be part of the way that this compound works. So as always, I always want to thank all of the people that were involved in this work. There are a number of pictures here, but not everybody I've talked about their work has been included. I tried to cite all of the papers so that you can have a look at them in your own time if you want to. So this is all research group, and I want to thank all of them and all the members of Serpell Lab that were with me before and are now. We get funding from the Alzheimer's Society, the BBSLC, the MRC, TauRx, as I mentioned, the Royal Society, Alzheimer's Research UK. And we work with Kevin Starras, Aidan Doherty, George Kemonet. I mentioned some of the work that we did with them. TauRx Therapeutics, the people that come from there, Charlie Harrington, particularly Claude Wishick, who is the lead of that company, John Story and Janet Rickard, and all the people that we've collaborated over the years. I mentioned some work from Joost and Fred, and also from Wei Feng Zhu during the talk today. And I'm going to leave you with an image of the University of Brighton. Is not actually the University of Brighton. So this is the seafront at the university, I would say in better times when there are people who are quite close together here. So this is the pier, and sometimes it's a stony beach, but sometimes the tide goes way out and we can see the sand, and it's, you know, a gorgeous day in the UK. Mahmoud also asked me to say something about myself, and so I do have just a few slides, but I'm aware of having gone on a little bit too long, so I might stop there, and then if people want to ask me questions, they can do so. So I think I'll do that.
All right, thank you so much, Louise, for the fantastic presentation. I think we do have some few questions, so we just jump through the questions. So the first question is actually from Simao Zulu. He said, please explain the link of E4 polymorphism with amyloid beta.
Oh my goodness. Well, that is a question that has an enormous answer, I'm afraid. So many people have shown that E4 affects the clearance of A-beta. Some people have shown that it affects the aggregation of A-beta. Some people have shown that it affects the production of A-beta. Some have shown that it affects the toxicity of A-beta. I mean, it really is an enormous subject and could be the subject of a very, very long talk. So unfortunately, I probably can't summarize that here, but there have been lots of studies that have suggested that having E4 has an influence on your response, I suppose, to amyloid beta toxicity.
Okay, so, and the next question is from Alexandro. He said, I'm curious about the usage of snails to evaluate the A-beta effect on memory. Why did you use this animal model?
Well, yes, so that is a very interesting question. So I work with George Kamenesh and Lindsay Ford, and they were two people who were working with water snails, so Limnaea stagnalis, to look at their memory. And it's quite interesting. I mean, this is not my subject area, but essentially you can teach the snails a task which is associated with feeding, and then you can measure their response to a feeding stimulus. So you can essentially measure their memory based on their feeding response. And so what we did was we used their assays, which is probably the subject of a longer discussion, to compare what happens in snails when you don't add A-beta or you inject A-beta into them. And what we found was that when you inject oligomeric A-beta, they actually forget what they've been taught. And then I wanted to mention it because in terms of the variant A-beta, it doesn't have that effect. So it was a nice control system. So snails are incredibly interesting and relatively simple organisms that potentially have real uses for understanding memory and learning.
Fantastic. Cool. So the next question is from Ajahn Lekoko. He said, does tau lead to amyloid beta deposition?
I think that one is quite a difficult answer. I would say that most people think that it might be the other way around. So amyloid beta tends to be observed. The oligomers of amyloid beta seem to be observed before tau. Many people have shown that amyloid beta aggregation may affect tau's behavior in some way. What's clear, of course, though, is that there are many diseases where tau is the main aggregating protein and not amyloid beta. So tau clearly has the potential to cause neurodegeneration by itself. It doesn't need amyloid beta to be there. But I think what happens in Alzheimer's disease, and as is shown by all of the genetic components, that amyloid beta does seem to be a very important sort of maybe a trigger of disease that then maybe leads to a cascade that involves the deposition of tau. I hope that makes sense. So I think it's the other way around, but I may be wrong. I think most people think it's the other way around.
Okay, fantastic. Okay, so the next question said, what is the native function of amyloid beta like tau that is known to support microtubules?
Another difficult question. Unfortunately, nobody knows what amyloid beta does. So some studies have suggested that amyloid beta actually is important in neuronal function. So you have a small amount of amyloid beta that maybe is important, but nobody has established a really clear role for amyloid beta, unfortunately. We still don't know what it does.
Cool. Okay, so we've got a question from Sylvester. He said, good talk. In our laboratory, we've been trying to model AD using A-beta peptide, but we struggle to get the peptide to become cytotoxic. Any guide on how to get this right?
Yes. So so the source of A-beta is really important. And the other thing that's really, really important is to make sure that you have removed any pre-aggregates. So as I showed in my talk, that if you add fibrillar A-beta to your cells, then it will not be cytotoxic. It's not very cytotoxic when it's fibrillar. So you need to make sure that you start off with something that is disaggregated. It needs to be soluble, it needs to be random coil, it needs to be non-aggregated, and it's only at the oligomeric stage that it seems to be cytotoxic. So it's hard to do that, but we use a method that involves a couple of solvents that we then remove to try and remove any pre-aggregated species. We're happy to help, actually, aren't we?
Cool. Yeah. So the next question is actually from my boss as well. He's the Secretary General of the Society of Neuroscientists of Africa, Professor Amadi. He said, have you investigated the role microglia may play in the protein misfolding as they do have an amyloid-facing pole or interactive surface where fibrils are said to be phagocytosized in the amyloid plaque?
Yes, that's a really interesting question. It's not something that we've done. People are looking at microglia and how they may be involved in phagocytosing amyloid. Fibrils may be involved in sort of spreading the amyloid load into different areas. We have focused almost entirely on neurons without really thinking too much about the other cells that are involved in the brain. And of course, that is a deficit. We need to think about what other cells like microglia, astrocytes, and so on might be playing. So unfortunately, I personally can't answer that question in this instance. So I apologize.
Okay. So we've got another question from Ibrahim Haruna. He said 3-O-heparin sulfate has been found to play important role in tau hyperphosphorylation. Is there any possible breakthrough in blocking 3-O-sulfur transferase enzymes?
I have to be honest, I don't know very much about this particular aspect. So I don't know if I can answer this without making something up. So I don't know the answer to that. I think hyperphosphorylation seems to be very important in the way that tau self-assembles. Having said that, what we've shown here is that we can use a truncated form of tau, and we don't need any phosphorylation, and we also don't need any heparin to get it to self-assemble. So how important hyperphosphorylation, how essential it is, I think still remains to be fully established.
Yep. So there is this question, but I think I may probably have to also give a background before. So it's from Chinna. She said, are there drugs that we can use, especially in Nigeria, to induce Alzheimer's animal model? And I probably think it's because most people do use different drugs. So for example, some people use aluminum chloride to try to induce some Alzheimer's like symptoms, etc. So yeah, I don't really know what you think of this question.
So I would say it's not my area of expertise, really, animal models. But what I know is that there have been lots of transgenic models that have been produced to try and recapitulate what happens in Alzheimer's disease. And I think that most people would agree that none of these are perfect. So I think it's possible that there could be some sort of toxin or drug that we could use to sort of induce some Alzheimer's-like pathology. But how do we do that so that it will reproduce and recapitulate Alzheimer's disease in a human? I think it's very, very difficult to do that. So I don't know of any drugs, and I think it's always hard to produce animal models that are identical to human beings.
Absolutely. Okay. So we've got this question from Amin Abdul Basir. He said, do you agree that targeting amyloid-beta might not help in ameliorating AD? If you do or not, can you say why? Do you believe targeting tau is better alternative?
Well, I think that a lot of companies and researchers have tried to target amyloid-beta. And so far, none of those drugs have worked. So some people would regard that as a failure, a complete failure, that suggests that amyloid-beta is not the right target. Having said that, it may be that targeting A-beta needs to be done much earlier in a person's lifetime. So we know that amyloid-beta starts to aggregate and form oligomers quite early on, long before we see any pathology. So if we were going to target amyloid-beta, we would need to do it before a person shows any symptoms whatsoever or any pathology. So the difficulty might be that there are drugs that would work, but we haven't been able to use them yet because we can't diagnose people early enough. The only way we might be able to do that is if we look at genetic populations, for example. So there's a group of people in Colombia who have a genetic form of Alzheimer's disease, and that was a place where I think many researchers were thinking that they could start trying to use some of these A-beta targeting drugs. So then to focus on tau, I think that the evidence does suggest that so far this drug that's tau-rx have that's in phase 3 clinical trials suggests that it may be a good target. It may be able to prevent Alzheimer's disease from progressing any further. I don't think we can ever sort of cure it, but once you treat people, then you might be able to halt it and stop it from getting any worse. I suppose if you think of amyloid-beta as being a trigger and then tau being a cascade, then if you're going to treat people when tau has already started to aggregate, then you actually probably are better targeting tau than you would A-beta, because the whole cascade has already happened. So that's my opinion. It may or may not be right, but that's sort of the way I think of it, that if A-beta has triggered tau to start aggregating, then we need to target tau at that point rather than trying to go to A-beta, because then tau has already gone off on its assembly pathway.
Cool. All right. So we've got this question from India. Dear Professor, thanks for updating our understanding of the disease, but I was more interested in knowing the mitochondrial dynamics associated with AD. Does it really matter?
I think mitochondrial dynamics are really, really important. We have looked at that, but not in a lot of detail. And I'm sorry that you were more interested in knowing about that, because, yeah, that wasn't. There are other people perhaps who would be better placed to explain mitochondrial dynamics.
Okay. All right. So let's see. We've got a couple of questions. Quite interesting. So we've got this question. So he said, since a strong pathophysiology has been established between AD and diabetes, could we start looking for drug candidates for hypoglycemic agents as drug repurposing concept? Yes,
I think that absolutely is a sensible plan. I think that there are a number of researchers who are looking into that. I personally don't work on that aspect myself, but I've looked at other papers where they've examined glycation of A-beta and looked at the connection between diabetes type 2 and AD, and there does seem to be a connection. So I think that it does seem to be a potentially exciting and possible route to target. Yeah.
Okay. Seems like a lot of people were quite fascinated by the presentation, lots of questions, which is good. So we've got another question from Amin. He said, regarding ApoEs, they are known to be involved in lipid toxicity in metabolic diseases. Is there a direct association between developing AD and disorders of lipid metabolism, like lipodystrophy or obesity?
Yes, so ApoEs are, of course, apolipoproteins. They are involved in lipid transport. I don't know of any connection between AD and lipid metabolism. Or obesity. I don't know if that's an extra risk factor or not. I'd have to look that up. So I don't know. Sorry.
All right. So we've got another question from the same person. Could AD on the one hand be a disease of lipid toxicity in the brain due to the role of ApoEs?
Again, yes. I really don't know. Certainly, from my point of view, I think the evidence is very clear that there's a very important role for protein misfolding. So whether lipid interactions are important there, I don't know. Of course, lipids are absolutely essential in terms of the membranes of the cells and so on, of the organelles. So lipids composition is going to be really important. But I think, I don't think that would necessarily be at the heart of it. I think at the heart of it is the protein misfolding. That's my view. Yeah.
So we've got a couple of questions, but I would first want to know whether you have a couple of extra minutes to spare first.
Yes, okay. I'm fine.
All right. Okay, so let's look at this next question. Chiku said, would mice produce similar results as snails, given that mice really do not suffer AD?
Yes, well, yes, that's a good question. So mice don't suffer AD, nor do snails. So I think, you know, we're always looking for a good animal model. We, you know, lots of people have also used fruit flies, Drosophila. Again, they don't suffer from Alzheimer's disease. So I think, you know, understanding this disease is really complicated. We can't actually study human beings, you know, in a molecular and cellular way. So we do have to use model animals, and we have to always consider that those are not going to perfectly recapitulate a human being, but, you know, they can give us some important and useful information to go on.
Okay, fantastic. So we've got another question, very generic. Joseph said, very nice talk, Louise. Considering our current understanding of AD pathogenesis and clinical manifestation, how close do you think we are to evolving potent therapeutic strategies?
Well, yes. Well, I mean, the work that I'm involved with TauRx is in phase 3 clinical trials. You know, if that passes, then I guess that's close. If it doesn't, then it isn't. I think in other neurodegenerative diseases, there has been some real progress in terms of gene silencing. So, for example, in Huntington's disease, and I know that researchers are looking into gene silencing or reducing production from genes as a potential therapeutic strategy. So, you know, we don't know, but I would hope in the next few years we will understand more. I think it's always hard to say how close are we. I don't know.
Okay. So we've got this last question, or at least second to the last question. So it's from Ismail. He said, very wonderful presentation. I noticed that most of the cell lines you've used for your studies are neuronal. What's your take on using cell lines such as hex cells to answer questions?
I think that using culture cells is really useful. In our own studies, we tend to use a cultured cell line called SH cells, so neuroblastoma cells. And then we moved on to primary neurons because we found that we got some criticisms about the fact that these were not real neurons. But then if you use primary neurons, then people will criticize you and say they're not human neurons and so on. So I think, you know, using cell lines such as hex cells and other cell lines is really important. I think that you have to often use more than one cell line to compare your findings in different cell lines. And you also have to consider that none of these models are going to be perfect. You know, people are now moving on to iPS cells, so human neuronal type cells, but again, they also have their limitations. So again, we're always looking for new model systems. None of it is going to ever be entirely perfect, but I think we can still learn quite a lot from using cell lines such as hex cells and neuroblastomas and so on. So, yeah. Okay.
So this, I promise, is second to the last question. So it's from James, actually.
Hi,
James.
Hello.
So he said, great talk, Louise. Have you developed a tau peptide that is scrambled like the A-beta one?
Yes. Well, no, we have not. But that's actually a really good idea. I should just say that it's not scrambled, and actually the A-beta, we have looked at A-beta scrambled, and it is not a good model. It's not a good control peptide. So we rationally designed our control peptide. I think it's a really good idea, and you know, so it tells us two things really, doesn't it? If we have a control peptide, it tells us which regions of the peptide are important for aggregation, and then it also allows us to use it in control in experiments. So so we need to know which part of the tau molecule we need to change in order to do that rationally. It's a really good idea, though.
Okay.
Thank
you. All right. So I guess this comes to my question. So I know that you haven't, because of time, you didn't present about your career, trajectory, etc. But I was wondering whether you could say a few things about, especially to ECRs, what advice looking at your career, would you give to early career researchers?
Yeah, okay. So the way that my career worked was, I must admit, not very strategic, not very well thought out or very well planned. I did a degree in biochemistry and genetics, and then I did my PhD on biophysics. And at the time, I didn't know really anything about structural biology. My biochemistry degree had not given me any background in structural biology at all. But I was really interested in amyloid structure. And so I really had to learn, and it was a very, very steep learning curve. So then I sort of became a structural biologist at that point. And I moved to Toronto, and I did some structural work there. And when I came back to the UK again, I started to realize that although structure was really important, trying to put it in the context of something that's more physiological seemed essential, really. And so that's why my group started to work on cell lines and cell biology and neuroscience. And I began to become more of a cell biologist and a neuroscientist than a structural biologist. But I'm never going to stop being a structural biologist. And I suppose the reason I'm telling this story is really to sort of encourage people to open up to different techniques and methods. So I think what I've always considered, the way that I want to sort of run my research, is to think, what is the question I want to ask and how will I answer it? And that means that you have to be very open to using lots of different techniques, not being bound by saying I'm a structural biologist and so I can't do any cell biology, or being a cell biologist and saying I can't do any structural biology. But you have to sort of look outside of your own field of interest and collaborate with others. So one of the slides that I had actually is a slide that shows how many collaborations. At conferences, I always try and talk to other people and get involved with new areas of expertise. In terms of setting up my group, I always found people who were going to come in and bring new expertise. Again, I mean, Mahmoud would be a perfect example of that he worked on tau in the cell, and that was very new for our group, and it meant that we then started to work on something entirely new. So you have to be very open to that. I think the other thing I really want to say, and I always want to say this, is how important it is to work with people that are supportive, that are interested in what you're doing, that want you to succeed, who are positive, and that works from me as well as to me. I'm very lucky that I have an excellent research group. They all work incredibly hard. They're all very supportive as well. And I try to give that back. So I think it's important to work in an environment that suits you, that makes you happy and also makes you creative.
Fantastic.
Does that answer some questions? I don't know.
Yeah, I think it definitely. So obviously, because we don't want to keep you for so long, I will have to end it here. And on behalf of the Secretary General of the Society of Neuroscientists of Africa and the members of SONA, we would like to thank you for giving this fantastic presentation. And of course, to all the audience, especially people that were watching from India and all across the world, thank you all for listening. Hopefully, we'll have you another time to give another lecture, or perhaps even come to Africa during one of these Society of Neuroscientists of Africa conference. Thank you.
Thank you very much. That was fantastic, and I'm honored to have had the chance to do this. So thank you all for listening.
Bye-bye.
Thank you.
Okay.